Namaskar, my dear students. Today we are discussing the second part of the chapter Mineral Nutrition, and in this chapter we shall be discussing the nitrogen metabolism. My dear students, apart from the carbon, hydrogen, and oxygen, nitrogen is the most prevalent element in the living organisms. Nitrogen is a constituent of the major uh, structural components of the plants such as amino acids, proteins, hormones, chlorophyll, and many of the vitamins. Plants, in fact, compete with the microbes for the limited nitrogen that is available in the soil. Here I must mention that nitrogen is the most abundant uh, element present in the atmosphere, which is about 78% of the nitrogen. But plants cannot utilize nitrogen in this way because they needed some conversion. Nitrogen therefore becomes a limiting nutrient for both natural and agricultural ecosystem. In fact, nitrogen exists as two nitrogen atoms joined by a very strong triple bond. This process, which is a process, is needed to convert it into some usable form. Plants cannot utilize the nitrogen in the atomic form. Some bacteria, blue-green algae, leguminous plants have the capacity to convert nitrogen into some usable form, which could be nitrates, it could be nitrites, and ammonia. Are amino acids in the soil. Organic nitrogen compounds in some insects are also the source of nitrogen. Now, the first and the foremost thing which is needed for the nitrogen to be available to the plant is the nitrogen fixation. The process of conversion of nitrogen to some usable form is called as the nitrogen fixation. It is the process that causes strong two-atom nitrogen molecules found in the atmosphere to break apart so that they can combine with other atoms. Nitrogen then gets fixed when it combines with oxygen or hydrogen, and generally it forms ammonia, which is the fixable form. Now, how does the fixation occurs? In nature, we have three methods by which the nitrogen can be fixed. One is the physical method. In the physical method, in nature, lightning and ultraviolet radiations provide enough energy to convert nitrogen to the nitrogen oxides NO, NO2 or N2O. Now in chemical nitrogen, there is next way is the chemical nitrogen fixation. The industrial combustion, forest fires, automobile exhaust, power generating stations are also the source of the atmospheric nitrogen oxides. Moreover, the use of fertilizers and the manures manually by the man in the fields is a source of chemical nitrogen which is available in the fixed form. The third and the most important source is the biological nitrogen fixation. Decomposition of organic nitrogen of dead plants and animals into ammonia is called as ammonification. Now, the biological nitrogen fixation that is available in the atmosphere due to the presence of the biological nitrogen fixation, it occurs due to the action of certain biological agents which could be bacteria of several types which causes the nitrogen cycle to occur. The nitrogen cycle wherein the atmospheric nitrogen first of all gets fixed either by the physical, biological or the chemical ways, and then it becomes available to the soil. The ammonia is first oxidized to nitrite by bacteria Nitrosomonas and Nitrococcus. The nitrite is further oxidized to nitrate with the help of bacterium Nitrobacter. These steps are called as nitrification. As you can see in this figure, the nitrifying bacteria are also called as the chemoautotrophs. The nitrate formed is absorbed by the plants and is transported to the leaves. In leaves, it is reduced to ammonia and finally forms the amine group of the amino acids. The nitrates present in the soil is also reduced to nitrogen, and this process is termed as denitrification. A special type of bacteria are available in the soil for the process of denitrification to occur, such as Pseudomonas and Thiobacillus. There are various types of the nitrogen fixations, and the most important is the symbiotic nitrogen fixation. Now, symbiotic nitrogen fixation occurs in the plants that harbor nitrogen fixing bacteria in their tissues. Several types of symbiotic biological nitrogen fixation associations are found. The most prominent among them is the legume bacteria relationship. Species of rod-shaped bacteria, bacillus type of bacteria, rhizobium, it has such relationship with the roots of legumes such as alfalfa, sweet clover, sweet pea, lentils, golden pea, broad bean, clover beans. In other words, with the leguminous plants of the family Leguminosae. The most common association on the roots is found in the form of nodules. These nodules are small outgrowths of the roots. The microbe Frankia also produce nitrogen fixing nodules on the roots of non-leguminous plants, and the example of non-leguminous plant where the nodules are formed is the Alnus. Both Rhizobium and Frankia are the free living in soil, but as symbionts they can fix the nitrogen. Now, if we uproot any one of the common pulse just before flowering, we will see a spherical outgrowths on the roots. These are the nodules. If we cut through them, we will notice that the central portion is red colored or either it is pink colored. If you imagine what makes the nodule pink, this is due to the presence of a special pigment called as the leg hemoglobin. And what is this leg hemoglobin? It is leg stands for legume and hemoglobin stands for a typical hemoglobin type pigment which has affinity for oxygen. Now, how does this occur? Let me discuss with you how does a nodule is formed in the plants. You can see in this figure, this portion, the first figure, this portion indicates the portion of the root. Nodule formation involves a sequence of multiple interactions between rhizobium and roots of the host plants. Principal stages in the nodule formation can be summarized in this figure. The rhizobia multiply and colonize the surrounding of the root. As you can see, the bacillus bacteria are surrounding the root, are get attached to the epidermal and root hair cells. The root hair curls, you can see in the second figure, and the bacteria invade the root hair just like this. Now, when this curl is formed and infection thread is produced, the infection thread, this green colored infection thread, this is the infection thread, green colored. This infection thread is produced carrying the bacteria into the cortex of the root, where they initiate the nodule formation in the cortex of the root. Thus, the bacteria are released from the thread into the cell which leads to the differentiation of specialized nitrogen fixing cell. Thus, the nodule is formed, establishes a direct vascular connection with the host for the exchange of nutrients. That's why this is symbiotic association, because both plant as well as the bacteria are going to benefit here. The nodule contains all the necessary biochemical components such as the enzyme nitrogenase leg hemoglobin. The enzyme nitrogenase is a molybdenum iron protein and catalyzes the conversion of atmospheric nitrogen to ammonia. Now, the ammonia is the first stable compound of the nitrogen fixation. The enzyme nitrogenase is highly sensitive to the molecular oxygen. It requires anaerobic conditions. The nodule's anaerobic condition means without the presence of the oxygen, that is called as anaerobic. The nodules have adaptations that ensures that the enzyme is protected from oxygen. To protect this enzyme, the nodule contains an oxygen scavenger called as the leg hemoglobin. As I just mentioned there, that this pigment is not actually the hemoglobin but has the role of similar of the affinity towards the oxygen. It is interesting to note that these microbes live as aerobes under free living conditions, but once they become symbiotic, they prefer to be in the anaerobic conditions. Now, this colony of the bacteria is formed, and this is how the this nitrogen, this legume, this nodule gets formed. Now, herein you can see the steps of conversion of the atmospheric nitrogen to ammonia by nitrogen enzyme complex found in the nitrogen fixing bacteria. Here in this figure, this is the enzyme nitrogenase, which combines with the atmospheric nitrogen, and immediately the triple bond between the two nitrogen atom gets broken because two hydrogen atoms get attached to them. Another bond breaks, and two more hydrogen atoms are attached formed. In this way, one nitrogen gets attached to three hydrogen to form the ammonia, and the release of the products occurs, and the enzyme nitrogenase gets free and can bind to another molecule of nitrogen. This nitrogen to form another molecule of ammonia. In this way, the nitrogen fixation occurs in the root nodule of the plant. Now, you must have noticed that the above reaction that the ammonia synthesizes synthesized by the nitrogenase requires a very high input of energy, and I must mention here that 8 ATP of the energy for each ammonia are produced. The energy required thus is obtained from the respiration of the plant, or the which is functioning as a host cell. Now, a question must arise in our mind: what is the fate of ammonia? The ammonia, at the physiological pH, the ammonia is protonated to form NH4 ammonium ion, while most of the plant can assimilate nitrate as well as ammonium ions. The latter, the ammonium, is quite toxic to the plant and hence cannot accumulate in them. Now, what happens to the ammonia? Because it is toxic, its fate proceeds in two ways. The first is the reductive animation. What happens in reductive animation? In this process, ammonia reacts with alpha ketoglutarate acid and forms glutamic acid as indicated here in this equation. And either if this does not occur, then the second way is the transamination. What happens here? It involves the transfer of amino group from one amino acid to keto group of a keto acid. Glutamic acid is the main amino acid from which the transfer of NH2, the amino group, takes place, and other amino acids are formed through transamination. The transamination enzyme transaminase catalyzes all these reactions as indicated here. Now I will explain my point with the help of this example, that the two most important amides, asparagine and glutamine, found in the plants, which forms the structural part of the protein, are formed from two amino acids, namely aspartic acid and glutamic acid respectively, by the addition of another amino group to each. The hydroxyl part of the acid is replaced by another NH2 radical. Since amides contain more nitrogen than in the amino acid, they are transported to other parts of the plant via xylem vessels. In addition, along with the transpiration stream, the nodules of some plants, for example soybean, export the fixed nitrogen as ureas. These compounds also have particularly high nitrogen to carbon ratio. This is how the nitrogen metabolism occurs in the plant, and we will talk about the nitrogen once again here, that because every element of the periodic table, as I told you in the first lecture, that is a part of the plant composition or the functioning or the transportation or the physiological biological phenomena, the physiology or the structure of the plant. But still nitrogen is discussed separately because it occupies a separate status. What is the separate status? It is abundantly available in the atmosphere but cannot be available to the plants unless and until it is fixed, and that is what forms the nitrogen metabolism. That's all for today, my dear students. Thank you very much. In case you have any query, you can call me on my phone number, or you can place a message at my email. And moreover, I would like to mention that you must go through the NCERT book for the more understanding. That